Okay, in this video, I'm gonna show you how to build a simulation that starts with a single cell and then we'll implement cell growth. And as the cells grow, we'll implement mitosis so that cells get divided into daughter cells. So I'm gonna start with clean slate. So essentially, I'm not gonna use any simulation. I'm gonna generate the simulation for you, just to walk you through the whole process. So to do that, I'm going to go to CC3 Python, CC3 project menu, click new CC3 project. I'm going to call my simulation mitosis. And here the simulation directory will be applications, CC3 4.2.3, demos, maybe my demos. I'm going to leave most of those settings as they are here. I'm going to introduce my cell type, call it condensing. There will be one cell type for now. I'm going to skip this part. I'm going to add adhesion energy, and I'm going to add volume local flex, and I'll explain what those guys are. And that's it. Hit continue, hit done, and we have our template of our simulation called mitosis ready. If I right-click it, open player, it should get open somewhere. There it is. Stop it again. I'm sorry. Pause it. The simulation should start running. So this is our initial pattern, right? That's not what we exactly want, because the simulation in this case starts with a rectangular slab of cells. We don't want it, so we're gonna do some modifications. I'm gonna open all the files of this project, go to mitosis.xml. This is where this initial cellular pattern is defined. This is this uniform initializer. We don't want it. So I'm going to delete this. And I'm going to save the file. Now, in the mitosis steppable, in the start function, I'm going to create first cell. So I'm going to CC3 Python menu, cell manipulation, create cell. Here I'm specifying the coordinates of the cell and its type. So for now we have only one cell type, and it's called condensing. So I capitalize all the letters of the word condensing, prepend self dot, and this construct gets resolved as a type ID for type name condensing. So if we look here in this XML specification, condensing will get resolved as type ID 1. If you run the project, I'm sure no, my computer is not open, so let me, sorry, the player is not open, so let me do this. And I wasn't fast enough. Let me step through the simulation. This is my cell. This is where it is, okay? And it disappears pretty quickly. The cell disappears pretty quickly because it does not have its target volume is set to 0 by default. So I will modify this cell a little bit. Modify the code first. cell equals. So what this statement does, it creates an object that stores my new cell, and the subsequent statement I'm assigning the pointer to my first cell to every element of the field between coordinates x coordinates 10 and 12 and y coordinates 10 and 12. So I'm laying out the cell, and I also will set lambda volume and target volume for this new cell. Equals 25. My lambda volume is equal to 2.0. Lambda volume, sorry. Now, if we run the simulation again, hopefully we should see a better picture. We don't. My cell still disappears. Let me make the lambda volume bigger. Yeah. So my constraint, my volume constraint, lambda volume was not strong enough, so I had to increase its value from 2 to 20. And now we can see that the cell survives. Alright, so now I will show you how to add a steppable that actually divides cells. So I'm going to select top level of my mitosis.cctd project. Actually, I have to be in this file, I believe. In steppable file. So expand the project, go to steppable file, right click, and select add steppable. And I'm going to call this steppable cell division. Alright? And here we can choose the frequency with which the steppable gets called. Let's call it every single Monte Carlo step. And the steppable type should be mitosis. So mitosis steppable is a special mitosis that contains additional code that is not present in the generic steppable, for example. So let's select that. Hit OK. And what happens is that Twedit generates cell division steppable for us. Okay? And it also modifies mitosis py, the main Python file, where we register the steppable with the main CompuSol runner. We can do some cleanups here if we want. Let's save both of those files. And let's run it, and let's see what happens. So if we keep running this simulation, nothing really happens because our cell, for whatever reason, does not divide. Well, let's see if we can figure out why the cell doesn't divide. Well, if we look at the cell division steppable code, we see this condition that says if cell volume greater than 50. Well, our cell volume will probably never reach 50. Why? Because our target volume for the first and only cell that we have in our simulation is set to 25. So it's highly unlikely that it will reach 50. What we have to do, really, in step function is to increase target volume by a certain amount. So let's do that. So in step function, instead of actually printing cell ID, I'm going to iterate over every cell in the simulation and increase cell dot target volume by 0.2 units. So target volume is a floating point number, just so you know. You can increase it in non-integer increments. Let's run the simulation and see if we see any change in behavior. So now, looks like we are getting somewhere. So if we zoom in, you can see that we are creating new cells. That's really interesting because the cells that we are creating have different colors. So what happened? What happened is that when cell gets divided, this function called update attributes gets called. And the parent cell, or the cell that existed before mitosis, gets split into two cells. So the cell object that existed before mitosis is called parent cell, and the cell object that gets created after mitosis is referred to as child cell. And here, what we are doing, we are actually assigning new type to the child cell, type 2. We don't want it. Why we don't want it? Because we have defined only one cell type of type condensing in our XML. So for now, let us modify the template and essentially differentiate the cell, the child cell. Save the simulation. Let's run it. So now this is what we should observe. We see more and more blue cells getting created because each of those cells grows independently of each other, and as they grow, as they reach doubling volume, which is set to 50, those cells divide. Right? So we'll leave it at that, and I'll see you in the next video.